Welcome to 17 Minutes of Science. My name is Sarah Cheeseman, and I'm a technical solutions scientist at Invivo Biosystems. We're glad to have you here with us today, and I am excited to talk about a topic near and dear to my heart, which is developing tools for zebrafish research. And my guest today is Dr. Raheel Samuel, and I will let him introduce himself in just a moment, but I'll tell you a bit about him. He has over 11 years of experience in developing microfluidic systems for many clients, and has made significant contributions in regard to high throughput systems for manipulating cells and organisms. So Raheel is actually an engineer, but he has kind of intersected with biology. So his work has benefited groups who are both academic types as well as commercial clients. And he's co-founded 2 biotech startups where he still serves in leading research and development efforts. He is the co-founder, one of the co-inventors of the ZEG, which is the zebrafish embryonic genotyper, which we will talk about today, that allows the ability to extract genetic material from live zebrafish, which is very cool. So that's what we're gonna talk about today. So I'm gonna let Raheel say a few words, and then we'll jump into the questions.
Yeah, hi. Thank you, Sarah. This is really a great privilege actually to come, and thank you for having me on this show. Basically, I would say in short, if I would describe my interests and professional pursuits, is coming with a microfluidics expertise to solve problems in life sciences that require more speed, more throughput, where samples are small, like zebrafish, and you can't solve these problems using other technologies where our technology can really contribute. So always enjoyed working with life scientists and making life better, healthier, solving problems with diseases, and zebrafish again has been doing that. So yeah, that's
great.
what
Thank you for that context. And actually, I want to jump into a question, but you, of course, and what you said just sparked another question, so I'm going to fold them together. But your background as an engineer, we'd love to know how you intersected with the zebrafish community. And I'm also hoping that you can fold in for people who are not so familiar with what microfluidics actually is, if you could give us a bit of a definition.
Yeah, sure. So microfluidics is actually the ability to manipulate fluids, any types of fluids, at a micro scale. So you get very interesting behaviors of fluids that you can manipulate to do very things that you can't do. Let's say you have a large amount of fluid. So it's very similar to what microcircuits are, or microelectronics is. Almost very similar technologies are used, except that in microchips you have current flowing through basically connection of circuits, but in microfluidics you have microchannels that you move fluids around. And one of the very popular application of microfluidics right now is single cell sequencing, high throughput single cells sequencing and manipulation. So it's and point of care systems things that you might have heard about, attachments that you can have on attached to your phone that measure something with a spit on sample or blood sample. They all use microfluidics. Inkjet printers are really one of the first applications of microfluidics, actually. So.
That's great background. So then how did you take that grounding and connect with zebrafish people and start working on their problems, which of course is fluid, all fluid.
Yeah. So one ideal thing zebrafish love, they live in fluids. They are in media all the time. That's a very good fit. The other thing that sparked my interest in them is I was working with C. elegans a little bit in my graduate school studies, and then I started looking around what other things are there and what other applications, other models that we can look at. And zebrafish was, I read about it and I saw that it's an upcoming model. I think that was in the 2000s, early 2000s. And we went to the university where I was going to. There were like 20 PIs at that school, University of Utah, and we met a few of them, and they numbered some of their problems that they have. And I think that's where the ZEG comes in, is you can actually, one of the hardest thing is to actually genotype these very small samples and keep them also alive at the same time. Can't be done with anything right now. So yeah, that's where the ZEG came in as an idea.
So it was really a collaboration between
it was, yeah, yes,
your engineering background and hearing what biologists were up against.
Yeah, so it was like really fun. Like they said, I mean, and this is the fun part about university research is like they are positioned to solve some very, very difficult problems that, let's say, in the industry won't take. And so you can have very good creativity like come in, and we worked with those on the people on the Zebrafish side, and then on, of course, on the engineering side. And then it's just fun to just, come up with, like, we couldn't even believe that people could, when we first came up with ZEG and when we first got a set of data, people were not like, it was like, oh, wow, can you actually do that? So that was fun to come to that point.
So let's talk about the ZEG some more, since this product has recently come to market. Can you tell us a bit about that journey from those ideation days that you're describing to where we are now and how long that's taken and some?
Yeah. Well, the way the light bulbs sound, you find out what doesn't work, right? So you do all these experiments. I think it's by the time it was like at least 3-4 years of trials, of experiments, trying different techniques, what is available. We had a group of, I would say, 6-7 people, including some PIs, some students, and then we had some like part-time full professional graduated engineers to work on this. And it was difficult, definitely, but I think what was the exciting part is the goal that we could actually, if we get this to work, the impact it could have. I think that kept us moving and, striving for that goal. So,
and how many versions of the ZEG existed prior to what's available today, do you think?
I would say at least 10 or 15.
Wow.
But you say, works, but it's not as good. So yeah, I would say at least that.
And then, so in that process of creating this instrument, which I do love the acronym, it's kind of catchy, the whole ZEG. I guess it's self-evident once you hear what it stands for. Yeah. You founded the company wFluidx. Did that come after you had the technology, or what were the steps there?
So what happened is when I first came, when I was in my graduate school, we started doing a little bit of experiments, which we saw what works, what doesn't work, and kind of get into a direction of what the possible methods could be for getting this done. We came across, actually, thanks to National Institute of Health, they released a solicitation that we want to make more tools for zebrafish researchers to make it more useful, make it help them adopt this model that could help in medical research. So we were in a great position at that time to apply for that grant, and that being a PhD student recent PhD graduate, it was a great opportunity to work on something that I have worked before, and also take a role of a PI and actually, build it to something that you can give it to others. So that was very, actually, I would say, like, National Institute of Health, definitely give credit to them. I think they're definitely behind. We needed that support to get there. So through that opportunity, when we did the more experiments and through that funding, we were able to launch a company, which is now, of course, commercializing it further on. So,
and how many people are now involved with wFluidx since things are rolling?
We are a growing company. Still, again, zebrafish is a very, very niche, space. We're small still. Probably, I would say at least 5 to 10 people, and we're growing. We just started, scaling up. and collaborating with other groups. So it's still pretty small, but yeah, the focus is to more expand our tools more and actually offer more opportunity for research for zebrafish. So
great. Well, let's go there because that's what I wanted to talk about as well.
yeah.
And so just briefly, for people who haven't had a chance to see the ZEG or have a visual of what it is and how it works, can you describe how somebody might use that in the lab and the problem that it's addressing for them in their workflows day to day?
Yeah, I think we try to fasten the pipeline of getting mutants to what they basically injection. You create a certain line, and then you have to raise them up, and then you have to crossbreed and everything. So they need it at a certain stage. We actually try to speed that up to help users to raise the fish raise only the embryos that they would like, because there could be, I mean, the hit rate is still small on terms of mutations, and so people have to spend a lot of time. So we're trying to almost cut down the time of raising the fish of interest from let's say 6 months or 3-4 months to in a month or a few weeks. That also helps in a way that some mutant embryos don't survive long time. They don't survive more than, let's say, 7 days. We actually are able to identify the mutants, let's say, in 2 or 3 days. So you can actually do that study that you want before 7 days, so that you can actually get that data that you want with 3 or 4 days of age embryos. So the ability of the ZEG can actually genotype 24 hours after the embryos, fertilized and stuff. So I think people have stretched, they've been able to genotype till 5 days. There are also other advantages in terms of like storing fish, again, so people are constricted with like fish tank space. So that actually helps there. You don't have to, again, raise that many fish. So you can get to your, with whatever resources you have, you can save on that resource of fish tank space. So
when I first heard about this instrument, that's immediately where my mind went, was just the huge ramifications for husbandry, where you rear a bunch of animals that ultimately are not useful to you. And by cutting that, over half those animals is a huge gain for the husbandry piece, and also efficiencies as you're describing. It's very cool.
So I mean, it really felt great that we can come in that part and fill that gap and help in that at that bottleneck. It was a very obvious bottleneck for a lot of people. So
yes, that's very true. So to continue on with that theme, you're thinking about tools of the future. Are you able to share with us any of the ideas that are percolating right now for what's next?
Yeah, that are brewing. I would say our goal is to enable Zebrafish as a high throughput model. That I would say broadly, we're trying to, and this is again, of course, coming around with the ZEG. One obvious next product is to increase its speed more, to actually add more automation to it. We're working on that right now. So along with that, basically our desire is to add more tools with that focus, so that we increase automation and also just try to get not the researchers out of the picture, but not in terms of tedious work. Get them thinking about their data, enable them to get the data faster, maybe with more automation, so that they can spend time planning the experiment, analyzing their data, rather than actually doing the tedious step to gather that data, and to automate it so it's more reproducible, repeatable. And I think that's a big need right now in zebrafish research, repeatability of procedures and protocols, as we have so far interacted with our customers. So what we have heard.
Yeah, I would echo that. And those are the kinds of conversations we have internally. And we also at NVivo talk to zebrafish researchers all over the world. And that is definitely top of mind. Automation will enable, the reproducibility piece is really good to think about that.
Yeah.
Okay.
Yeah.
We have a couple minutes left. So I have a timer started, and I failed to tell you I started it, but
Mhm.
I know you know I was doing that. I wanted to turn to just a related topic, but away from the ZEG, and that is that you are an entrepreneur. And I think it's always interesting to hear people's stories. You said something to me when we were chatting before we were live about your interest and what drives you in research for now versus research for 10 years down the road. I think you could say a little bit more about how you became an entrepreneur and that vision that you have for the now.
Yeah, yeah. I think so. To be honest, is like when you are part of something that could actually, as a scientist, you discover something that can help science, benefit the growth of science or even benefit health and stuff like that, which where I was in with the ZEG. It was like, wow, you can let's work on that and keep on, like you're in such a great position to make it grow. I think as scientists, that's where I came in as like, okay, can I be with this for a more longer? Can I take it to a stage where it can be adopted quickly and can be adopted in a wider audience? Because right now it's just sitting in this lab. So I think every graduate student who's working on applied research or science, they might have that in them. And I think I would encourage you that, I mean, we have the support, again, as I mentioned, from National Institute of Health and also other agencies, I'm sure, that can help you, get to take your idea to at least on the commercialization scale, because entrepreneurs are so important. I mean, you can have the best ideas in the universities and very in these idea creation centers, but you need those people. And there are different types of entrepreneurs. Like some people are very good at scaling businesses, managing business, taking the risk, and there are all kinds of them. So as a scientist, you can be that entrepreneur that can actually, the you can that will be your baby, what I would say, your idea that you make it grow and then share it with others. So I think that was my motivation mostly that brought me here. I actually never thought, by the way, that I'll be doing this. So
I think a lot of people say that.
so yeah, it's very I think the most the biggest thing was just, oh wow, this technology can help others. Yeah, I mean, and that's great. That's a great drive, and don't be afraid, I guess. So go for it. So
Those are good words of advice. And so in our last 20 seconds, if you looked back 3 years as a leader of your company, what would you tell yourself that you know now that you didn't know then?
oh, that's hard to, like 20 years from now.
No, if you look back 3 years ago from today at how things were, what you know now and how things were then, what would you tell yourself?
I would say just encourage myself, I guess. You never know what you're going to see. I'm surprised so far. And I think if we are willing to take the risk and willing to work hard on something we believe, I think it's worth it. And I would say, I think startups and it's such a great opportunity to learn so much. And I would encourage, I was not sure at that time, but I would tell myself, hey, no, this would be good. Go for it.
That's a great way to end. And our timer went. So don't let fear hold you back.
So,
Raheel, this was really fun. Thank you so much for
thank you, sir.
about the journey of the ZEG and to finding Microfluidics, which of course sounds like in the age of COVID is relevant every single day with every test that people are taking and analyzing. So we look forward to hearing more about where wFluidx goes in the future and wish you all the best.
Yeah, thank you so much for having me. It was a pleasure.
Well, thanks everyone for tuning in, and we'll see you next time.